Good morning. So I'd like to begin my talk this morning with the following question. Is there anything morally wrong with artificially creating a human brain for medical or research purposes? You might think that's a question for science fiction novelists. However, new bioengineering research makes it possible to create self-assembling multicellular systems, such as organ-like structures and biorobots, from a variety of cell sources. Engineering multicellular systems is a rapidly advancing field within which new categories of engineered living systems raises ethical and societal questions regarding responsible innovation. For the past year, I've been fascinated with the broader impacts of engineered multicellular systems, and today I'm happy to be speaking with you about this constantly evolving topic. In this talk, I'm going to focus on brain organoids, which are also called mini-brains. I will begin with what they are and how they are used. Then I will turn to the question of whether it is morally permissible to engage in growing mini-brains. I will argue that given the current information that we have, engaging within this field of research raises grave ethical concern. I will approach the moral question through two separate arguments, the first through consciousness and personhood, second from the approach of personal identity. Finally, I will point towards the nuances within this issue and how further research could lead to a different answer regarding the ethical nature of conducting human brain organoid research. I hope to introduce this topic as a new ethical puzzle that challenges our current understanding of the categories of living systems, particularly those built out of human cell types. To begin, organoids are multicellular systems that resemble organs at the early stages of embryonic development. For example, human brain organoids look like these pictured here on this slide. Just so you know, this is the only slide that I have today, so we can keep our eye on what we're talking about. Here, you can see that the organoid is organized in the same way as an actual embryonic brain. You can see the neural stem cells in red and the neurons in green. You can see that the organoid and the embryonic brain are identical in their organization. However, organoids currently grow to only about 4 millimeters in diameter due to lack of vascularization. These organoids are grown in a bioreactor from human pluripotent stem cells. They aggregate and differentiate and then develop structural components of the brain, including regions reminiscent of the cerebral cortex, the choroid plexus, the retina, and the meninges. All that is to say that the organoids mimic mechanisms of both mammalian neurodevelopment and more specifically human brain development on a small scale. The significance of brain organoid research is that we will be able to study development, being able to directly measure structural changes under conditions of health and disease. Researchers hope to use brain organoids to study a plethora of neurological diseases, including Alzheimer's and autism, as well as mental illness. The promise of brain organoid research is to reveal how our brains and possibly our minds work. As previously mentioned, currently organoids are limited in their ability to grow based on size limitations due to lack of vascularization, which us tissue engineers call plumbing issues. This is a central research area for a variety of ex vivo cell systems, including brain organoids. And advances within tissue patterning, such as lobe formation, have accelerated research in this field. Most importantly for our discussion today, brains self-organize in the same way that brains self-organize during embryonic development. So organoid research is already providing scientific insight to brain development, plugging knowledge gaps relating to understanding developmental organization of the cells within the brain. As tools are developed to assist the growth of cerebral organoids, the complexity of these organoids increases and more functionality develops. That is to say that organoids start behaving like their embodied counterparts. This means that us scientists must now face ethical questions associated with the engineering of complex artificial human organs, in particular, the creation and manipulation of brain matter, which is central to human identity, and this raises concerns about the respect for human dignity. Human brain organoid research is both scientifically and morally new and must be approached with new arguments. It might seem at first that the ethical concerns regarding organoid research have already been addressed by the discussions pertaining to embryonic stem cell research or cloning. Unlike embryonic stem cell research, organoid research does not entail the destruction of an embryo to harvest the cells. Organoids are made from adult stem cells. Perhaps we might think that the same ethical analysis of cloning might also apply to brain organoids, since brains are a significant part of the human person. However, organoid research does not aim at creating a fully functioning human being. Thus, the human brain organoid requires new scientific and moral evaluation. Here, I'm going to explore two new resolutions to my central question, which is it ethical to create and conduct research on human brain organoids? First, I will examine this question in light of some considerations regarding consciousness and personhood. To begin, philosophers of the mind raise concern of consciousness when considering the ethical nature of research in this topic. A conscious thing has perception and perspective, and a rational thing can see itself as the object or subject of an action. Such psychological phenomena allow something to see itself as a subject of wrongdoing, which leads us to generally consider it wrong to harm that thing. So, at what stage of structural complexity do we consider a human brain organoid similar enough to an actual brain? Moreover, at what stage can it be considered a consciousness-supporting brain? And then, how do we establish when the organoid has become conscious? Finally, when can we detect the presence of rational thought? These questions are important because together we want to know if having all of the structural components of the brain is sufficient for supporting human thought and therefore establishing moral status. Asking if organ-level organization of a brain is sufficient for rational thought gets us into philosophy of the mind territory. We don't have the scientific tools to answer questions of the mind-brain connection. And even if we can answer the question of when a multicellular system becomes a brain, neurobiologists are in no position to answer the question of when the brain can support consciousness. This is precisely the problem, that our knowledge about ourselves begins to fail, and that is, we have no idea how our brain is actually connected to the mind. We understand the biology of neurons and synapses, but there is a key scaling issue that neuroscientists and bioengineers are now tackling. There are several key missing links between a neuron or a set of neurons firing to a thought or a feeling. Thus, organoid research is exciting because it raises questions at the core of what it means to be human. However, given that we cannot currently empirically determine what about our brains gives rise to conscious experience, or whether it is the structure of the brain alone that gives rise to conscious experience, why should we concern ourselves with the ethical treatment of organoids? Because what we are dealing with, the brain and the mind, is importantly related to moral status, i.e. personhood, dignity, and identity. Therefore, if we are unsure of what we are dealing with, then we should be cautious about how to proceed. Rather than being reckless, the best course of action is to assume that organoids are conscious, then we should be cautious about how to proceed. It is important to acknowledge that our mind is likely linked to what separates us humans from other animals. If an organoid could have awareness, there are additional ethical concerns regarding pain sensation, phantom body, or even locked-in syndrome. If the organoid could support consciousness, would it have rights? The capacity for rational thought supported by brain function is the basis for moral status under nearly any philosophical framework. Moral status afforded to rational creatures requires caution regarding the manipulation and destruction of the thing created. From the Catholic perspective, since it is unclear what we are creating when we produce a brain organoid, the mere probability that something like a person is being created prohibits brain organoid creation. Here, I'm going to draw on church teaching within Evangelium Vitae and Dignitas Personae. Within these two documents, we can draw from similar situations such as therapeutic cloning. Therapeutic cloning occurs when embryonic stem cells are produced without the destruction of a true human embryo. For example, the development of an unfertilized egg. This technique raises questions on both a scientific and ethical nature regarding the ontological status of the embryo-like product obtained in such a way. Until the ontological doubts of said embryo-like product have been clarified, Evangelium Vitae tells us that what is at stake is so important from the standpoint of moral obligation, the mere probability that a human person is involved would suffice to justify an absolute clear prohibition of any intervention aimed at killing a human embryo. Furthermore, bioethicists Ashley and O'Rourke remind us that God calls humankind to join with him to share in his creative power through the gift of intelligence. Principles of stewardship and creativity call us to be genuine co-workers with God in an evolutionary process which is not yet complete. And within that, we are encouraged to exercise real originality. They go on to say that it is important to remember that human creativity depends on a human brain, and the activity of the brain depends on the emotions and the biology of the body. Any alteration that would injure the brain, and hence a person's very creativity, would indeed be disastrous mutilation. Alterations that reduce a person's ability to sense the world or to experience emotion are contrary to the principle of totality, i.e., the good of the entire person, because emotional life is closely related to human intelligence and creativity. To summarize, principles concerning consciousness and personhood for growing and engineering the home to our intellect raise concern regarding the moral status of brain organoids. Thus, we must conclude that human brain organoid research raises grave ethical concerns. Having established that brain organoids should not be created due to concerns of consciousness and personhood, I will now add a further reason why these organoids should not be created. I will talk about personal identity and the relationship of the brain thereto. Each person's brain is central to their personal identity, thus recreating their brain outside of the body is an affront to personal identity. But what is identity? The English word identity comes from the Latin word identitas, which is a combination of the word idem, meaning same, and the suffix itas, meaning ness. In other words, identity means sameness. A thing's identity is simply a phrase that identifies a thing as being this particular thing. It is widely considered unethical to transplant a face because a face is specifically tied to the identity of that particular person. We identify people by their faces. It is necessary to note that facial tissue grafts for burn victims is allowed because it is an attempt to restore the function of the facial skin barrier. Brains, like faces, are specifically tied to one's personal identity. They are specifically tied to how we see ourselves. Self-image and identity are reflected in our corporality. Embodiment is integral to the person. Thus, the preservation of our particular identity is important because it is necessary for wholeness. A person is a unity of body and soul which possesses a mind. Getting back to the example of brain organoids, functional integrity is necessary for corporal integrity. Can a brain that is disconnected from a human body preserve functional integrity? Stepping out to assess the functional integration of body and mind, a brain outside the body lacks a connection to a key component of the person, its body. Because the identity of a person is tied to their brain, it is important to ethically protect that personal identity. Thus, culturing a brain ex vivo is not morally permissible under a personalist worldview. To summarize, human dignity requires preservation of identity, and our brains are centrally linked to our personal identity, and thus require protection from transplantation or ex vivo culture. In thinking about the tissue engineering of multicellular systems with increasing complexity, there seems to be a spectrum where on one side we have cell culture, which requires some ethical protections. The protection of personal information comes to mind. When we culture primary cell lines, we must first de-identify the samples to protect the donor. Whereas on the other side of the spectrum, there's research concerning distinct people, which require ethical considerations regarding rights. Consent here is a big concern, and preservation of human dignity, i.e., do no harm. Within this spectrum, somewhere between a whole person and a petri dish of cells, lies organoids. Thus, we are inclined to ask, at what level of organization of a multicellular system does moral status arise? Brain organoids are made with adult stem cells, which present little ethical concern. Furthermore, an embryo is not being created, there is no conception, so likely there isn't the creation of a soul. Without a soul, likely there is no rational mind. So taken together, there may be conditions under which brain organoid research may be morally justifiable. For this to be true, we would have to assume that the mind-brain connection only exists if a brain is grown inside a body. Here, we have to unlink the brain from personal identity, and if anyone knows the experiment or the thought experiment that would unlink these two things, please let me know. Under the assumption that human brain organoids do not contain minds, then there can be only computation, like a mouse's brain, and not thought. Philosophers of the mind and artificial intelligence experts have been trying to resolve this one for a while. We continue to fail to describe the mind using scientific tools. And given a better understanding of the mind-brain connection, there could be a scenario in which brain organoid research is morally justifiable. However, with our current understanding of the person, we are required to protect human dignity, not splitting the mind from the body. To summarize, the current state of understanding of the person and the human mind-brain connection requires specific moral principles protecting the dignity of all people. Consciousness assessment requires additional scientific research to elucidate what makes a human brain different from other multicellular systems. The fact that the brain is centrally linked to personhood further requires a specific ethical protection to maintain human dignity. Furthermore, in protecting the integrity and identity of a person, the brain requires special protection from transplantation or explant. Since organoids could be considered three-dimensional cell culture, there could be a spectrum within which research on human brain organoids would be allowable to a point. In considering these points regarding the ethical nature of cerebral organoid research, it is essential to regard the real meaning of being human. Beyond disease-free biological function, wellness involves the integration of biological, psychological, social, and spiritual creative needs. To conclude, I want to thank you for your attention and leave you with an excerpt from Ethics of Healthcare by Ashley and O'Rourke. Natural law should not be conceived of as a fixed pattern of human life to which human beings are forever confined. Rather, the Creator has made human beings free and intelligent, and it is precisely this intelligent freedom that is human nature and the foundation of moral law. Human intelligence, however, is not disembodied. It depends on a brain and a body that have a specific structure. In caring for their total health, persons not only have the right, but the obligation to understand their psychological and biological structure and to improve themselves, even in ways that may seem novel to past generations. Such improvements are good stewardship and share in the divine creativity with which God has endowed humankind. And with that, I would like to open up the conversation to all of you. What do you think? Is there anything morally wrong with artificially creating a human brain for medical or research purposes? Thank you.